All right. So we're now going to move from the um, non-specific to the specific. So we're now going to talk about the immune system, but the acquired part. Okay? So in the acquired immune system, uh, we are going to emphasize a few things. The first thing we're going to emphasize is that there is learning that takes place, learning about the pathogen. Aside that, there is storage of the information, okay? The storage of that information, and then the information is used to create defense, okay? So there is creation of defense, okay? And this, we must know that the building or the creation starts immediately after birth. So we see that as you grow, you get to build defenses against various types of pathogens, bacteria, and other harmful proteins or substances. So just a reminder, antigens, these are body antibody generators, okay, and they cause the body to identify a pathogen and create an antibody against it. All right. So let's just go through some names, some terms rather. So we have antigen. We want to remember that these are antibody generators. So if you have a pathogen, this would be the antigens. And this are the ones that are going to help create antibodies. And these antibodies are the ones that are going to help fight against these pathogens. And by fight, I just mean, first of all, we're going to have recognition, okay? Recognition of the pathogens, and then they're surrounded, and then chemicals are released by nearby phagocytes, and it's a mechanism that we will talk about um, in this video. So to start with, we will look at the cell-mediated response. So in the cell-mediated response, we are mainly looking at the involvement of T cells. Now, there are different types of T cells, and these include helper T cells, okay, and then cytotoxic T cells as well. The helper T cells, uh, these activate and direct cells that can kill the pathogens. So these are activators. They're activators, and then they can also direct uh, cells that can kill the pathogens. So in the next few slides, we're going to appreciate what the role of these T cells are in the next few minutes. So we ended on antigen presentation. So let's say we have a dendritic cell that has just presented, okay, on the MHC2. All right. So first of all, we have the phagocyte. We have the phagocyte, which is the dendritic cell. In this case, it engulfs a pathogen, and then parts of the bacteria or parts of the pathogen goes to the surface of the phagocyte, which is what we have done here. So the phagocyte, after presenting, will be recognized by a T cell. So I'll put my T cell in blue, all right? So what will recognize is actually the helper T cell. So the helper T cell will recognize what is happening here and will be attracted to this particular phagocyte. And once it attaches, all right, I'll just draw this time so that we're able to attach them. Once it attaches, so this is a presentation on the MHC2, we will have the T helper cell, the helper T cell, attach, and once the attachment occurs, right, so there will be presentation, and then there will be attraction, then there will be binding. After binding, the T helper cell will be activated. So what do we mean when we say activated? What happens next? So in this case, we've seen that there's attachment, the T cell becomes activated, then what? All right? So what happens now is that they make copies of themselves, all right? So the T helper cell will make copies of themselves, and then they will differentiate. All right? So they will make copies, and then after making copies, they're going to differentiate. Differentiate into what? All right? So we have effector T cells, and then we have memory T cells. All right? So the effector T cells, what do? The effector T cells travel around, they move around, signaling to other lymphocytes that there is an invasion that has taken place and that we need some action to take place. All right? So how do they do that? They do that by releasing cytokines. You remember these guys? Yeah. What do the memory T cells do? So the memory T cells, they keep a record of the intruder. All right? They keep a record of the intruder for future attacks. So if the intruder was what we saw in here, they're going to keep, and if the intruder, let's say, was Teresa, they're going to keep a record of Teresa so that once Teresa comes a second time, they will identify to say Teresa is not part of us. So in this case, when the body is exposed to Teresa again for the second time, the memory T cells will identify Teresa, and they will immediately activate the T cells so that the invading Teresa is destroyed quickly. Okay, in this case, there's no need for the phagocytes. They will just go and Teresa has come, let's go and attack. All right, so the response of the T cells in this case will be more powerful the second time because they will know that Teresa is not part of us. In the first time, it took someone to identify Teresa. After identifying Teresa, they swallowed Teresa up, put the structure of Teresa on the surface of the phagocyte, and then that is when we had the T cell come. After coming and attaching to the Teresa peptides, then it made effectors and memory T cells. The memory T cells then had the Teresa structure in them, so that the next time that Teresa comes in, they just pounce and then have the destruction happen quickly. Okay, so that is what the T cells do. I'm just going to talk about the cytotoxic T cells a little bit. So the cytotoxic T cells work in this way. When the cells are severely infected, so we have a cell and it's infected severely. We have it presenting an antigen on MHC1. We remember this. We remember this story, right? So the cytotoxic T cell, they're able to attach, okay, and then also kill these cells. All right. So we have an infected cell, and then an antigen fragment is placed on MHC1, and when it is placed on MHC1, then we find that, so this is the presentation, then we see that the T cell, in this case the cytotoxic T cell, is able to identify that. So the combination of the MHC molecule and the antigen in this case is recognized, okay? And once it is recognized, then it is able to ensure that the infected cell is killed, okay? So the cytotoxic T cell will inject, all right? So it will first of all recognize. Once it recognizes, then it will inject. It will inject in particular lysosomal enzymes, and then we'll see that this infected cell is killed. This is different from what we talked about in the NKC. Can you pause the video and see what happens in the NKC and how it is different from this one? So just note that all nucleated cells have MHC1 presented on the membrane, and the infected cell will present parts of the proteins on the MHC1, and this activates the cytotoxic T cell for lysosomal action. So kindly state the differences between NKC and cytotoxic T cell mechanism. In the next one, we'll be talking about the humoral component of the acquired immunity.